Hello everyone, welcome back to my channel Microbiology Simplified. In this video PYQ series, I'll be discussing about Nocardia. This is very important bacteria because it is known to cause a variety of infection, and some of the infection resemble other diseases such as tuberculosis. So without wasting any time, let's start with the video. The topic is Nocardiosis PYQ number 13. We'll start with the MCQ. It is about a patient who presented with a history of fever, cough, weight loss and anorexia. Microscopic examination of ZN stain smears of his sputum was negative for microbial tuberculosis. Chest X-ray shows pneumonia. BAL fluid was examined microscopically, which showed gram-positive branching filaments, and it was partially acid-fast. So what is the most likely organism? Also the image is given in which we can see the gram-positive branching filamentous rod. Okay, this gram-positive, this branching we can see here, and also we can see so many filaments overall. Okay, so apart from that, what other information given the question is the presentation is of pneumonia with fever, cough, weight loss, anorexia, and also the chest X-ray suggests pneumonia, but the ZN stain is negative for Mycobacterium tuberculosis, and the BAL fluid shows gram-positive branching filament, and also it is partially acid fast. So we'll see the options. The first option, Mycobacterium tuberculosis, is excluded because the ZN stain is negative for Mycobacterium tuberculosis. Also the last option, this Mycobacterium leprae, which is known to cause leprosy, is excluded because it does not involve lung and does not cause pneumonia. Now the second and third option, if you see, this Nocardia, Stauroides and Actinomyces, both are gram-positive branching filaments, but then Actinomyces israeli is non-acid fast. So that's why the answer to this question is Nocardia stauroides. So apart from this, there are certain other more information which we should know, because in examination the same question will not be asked. So we'll go into the precise detail about Nocardia so that you will be able to answer all the questions from this topic. So let's start. So as I told you, it resembles morphologically being gram-positive branching filament with Actinomyces israeli, but there are certain difference between these two, and what are those? Nocardia species are strict aerobes, whereas Actinomyces israeli are anaerobes. If you remember the last video of actinomycosis, are anaerobes. Okay. It is weakly acid fast, whereas Actinomyces israeli is non-acid fast. And also the infection which we see in case of Nocardia is exogenous, whereas in Actinomyces israeli it is endogenous. If you remember the previous video question, the patient underwent dental extraction before the infection, so the infection was endogenous in origin, whereas in case of Nocardia it is exogenous. So as it is strict aerobe, so it can reside freely in soil, organic matter and water, and this it is weakly acid fast. It is because of presence of obviously the mycolic acid. Apart from mycolic acid, there are certain structure which are present which is specific to Nocardia, or we can say a group of organism which resembles Nocardia. And what are those structures? Those are mesodiaminopimelic acid, in short we called it mesodap, and arabinogalactan. Okay, so if you see the structure, this mesodap is present in the peptidoglycan layer. Basically this mesodap is responsible for cross-linking of peptidoglycan layer, and this arabinogalactan layer is present above the peptidoglycan layer, and next to it is the mycolic acid layer, and then the top layer which is consists of glycolipids and various other proteins. So this structure is not only specific to Nocardia but to a group of organisms which we called aerobic nocardioform actinomycetes. Aerobic nocardioform actinomycetes. And what are those organisms? Those are Mycobacteria, Corynebacteria, Nocardia, Rhodococcus, Gordonia and Sucamurella. So all these organisms basically belongs to an order known as Mycobacteriales. Okay, known as Mycobacteriales. So all of them have got this peptidoglycan layer, in which we can see this mesodap, then followed by arabinogalactone, and then mycolic acid. The only difference between is the composition of these structure. Basically, they vary mostly in the composition of mycolic acid, that is what is the thickness of mycolic acid, number of carbon atom in the mycolic acid. So it is highest in Mycobacterium, followed by Nocardium, and least in Corynebacterium. Okay, so that is why Corynebacterium is non-acid fast, whereas Nocardia is weak acid fast, Mycobacterium is acid fast. Okay, and also as it can reside in soil, organic matter and water, so that's why the infection is exogenous. So these are the differences between actinomyces and Nocardia. Now coming to the various species of Nocardia causing infection. So the most common species are Nocardia steroides, Nocardia brasiliensis and Nocardia otitidis caviarum. So initially thought that Nocardia steroides is a single species, but later on it's found to be a complex of different species based on the drug sensitivity pattern. So these are called as Nocardia steroides complex, and these various species which belongs to this complex are called as drug resistant types, and these types are type 1, type 2, type 3, type 4, type 5 and type 6. And this type 1, the species is Nocardia abscessus, type 2 is Nocardia brevicatena, type 3 is Nocardia nova, type 4 is Nocardia transvalensis, type 5 is Nocardia farcinica, and type 6 is Nocardia C. georgica. See, some of these type or some of these species are themselves are complex and contain various other species. So among these total 6 types of Nocardia species, this Nocardia C. georgica is a very commonly infection causing species, and it is usually associated with pulmonary infection, whereas this Nocardia brasiliensis and Nocardia otitidis caviarum is usually associated with cutaneous infection. Okay, and the clinical form which we can see in Nocardia, or the clinical disease or the disease which we can see, are basically of 3 types. First is subcutaneous, second is pulmonary, and third is disseminated infection. And among these 3, the predominant manifestation is pulmonary. Pulmonary is predominant clinical manifestation of nocardial infection. Okay, so we'll see one by one. This subcutaneous manifestation, it usually follows traumatic transcutaneous inoculation of nocardia, which may be present in soil or organic matter. Okay, through minor trauma it will enter into the skin, and what it will lead to? It will lead to 3 types of disease. The first one which you can see in this image is cellulitis. Okay, so cellulitis, it starts with a warm swelling. Later on it will develop into a firm non-fluctuant nodule, and it may progress to involve underlying muscles, tendons, bones or joints. If you remember, in case of actinomycosis, it also has got that ability to progress through various tissue plane. Similar to that, nocardia can also progress to various tissue plane and involve muscle, tendon, bones or joints. So this is the characteristic of both the actinomycetes. Okay, and the species which is commonly involved, as I told you, is Nocardia brasiliensis. The second infection which we can see in this image is lymphocutaneous nocardiosis. In this, at the site of inoculation, we can see a pyodermatous nodule, and along the draining lymphatic, what we can see subcutaneous nodules. You can see this subcutaneous nodule along the draining lymphatics. Okay, and this resemble infection caused by Sporothrix skenki and Mycobacterium marinum. Okay, and again the species which is commonly responsible for this lymphocutaneous nocardiosis is Nocardia brasiliensis. Okay, apart from these two, the third disease which we can see commonly caused by nocardia is actinomycetoma. Actinomycetoma. For actinomycetoma, I have made another video separately to distinguish it with eumycetoma, which has got similar clinical features but then different etiology. So for that you go through that video, okay, and the link is mentioned here. Now the second clinical form, which is I told you the predominant, is pulmonary manifestation or pulmonary nocardiosis. And what we can see, pulmonary nocardiosis is common among adult males and among immunodeficient individuals. And the diseases such as AIDS, transplantation and steroid therapy is responsible for that immunodeficient status. Among AIDS patient, it usually occurs when the CD4 cell count is less than 200 per millimeter cube. Okay, and how it occurs? It usually follows inhalation of contaminated aerosolized droplets. So aerosol inhalation, aerosol droplet inhalation is the main source of infection. And what else we can see? This pulmonary infection or pulmonary manifestation is usually present as pneumonia, and it is typically subacute, okay, and it present with fever, anorexia, weight loss and malaise similar to pulmonary tuberculosis. But the lesion is usually suppurative, but it may be granulomatous as in case of pulmonary tuberculosis, or mixed, it is both suppurative and granulomatous. And on X-ray or CT scan, what we can find? We can find infiltrates or nodules. These nodules may be single or may be multiple. We can find cavities then which resemble TB, or we can find abscess, okay. And this, as I told you, it has got a capability to progress through various tissue planes. So contiguous extension may be seen to surface or deeper tissue. So it may go either toward the surface or toward the deeper tissue, okay. So this is the characteristic of pulmonary nocardiosis, and the species which is commonly involved in pulmonary nocardiosis is Nocardia steroides complex. And if you remember the species, Nocardia cyresi georgica, okay. So and it leads to approximately 90% pulmonary nocardiosis. Now the third clinical form which we see is disseminated, and where it can disseminate, it may disseminate to brain, which is the most common site for dissemination, okay, followed by eye, where it can most commonly involve retina, or it may involve the skin and subcutaneous tissues or kidney. And what is the most common clinical manifestation? The clinical manifestation is commonly seen in the disseminated nocardiosis is, which you can see in this image, is the subacute abscess, okay. So these are the various clinical forms which we can see in nocardiosis. Now coming to the lab diagnosis. So diagnosis can be either made by microscopic examination or by the culture of the specimen. So microscopically we can examine and visualize the organism via Gram stain or via Zn stain as it is weakly acid fast. So what is the method which we will use for this weakly acid fast bacteria? Modified Cugnon's acid fast stain. And what is the difference? This modified Cugnon acid fast stain, we will use 1% H2SO4 instead of 20% H2SO4 which we use for Mycobacterium tuberculosis. And what we will see in this both the stain, we will see the beaded appearance. You can see here this beaded appearance, and then this filaments, you can see the branching filaments, and in Gram staining you can see the Gram positive filaments, and Zn staining you can see acid fast branching filaments. So the characteristic is beaded branching filamentous rod, okay. And on culture, if you see, the growth is very slow, and it usually takes up to 2 weeks to grow, but commonly it will grow in 3 to 5 days, okay. And it can grow in non-selective media, but if we are culturing it from a specimen containing mixed flora, so we should use selective medium. And what are selective medium which we can use? There are no certain specific selective medium for Nocardia, but selective medium for other organism can be used for it, like Thayer-Martin medium, which is used for which organism? Neisseria. And we can use paraffin agar, or we can use this buffered charcoal yeast extract agar, which is used for Legionella. So this paraffin agar, basically this technique of using paraffin for cultivation of Nocardia is known as paraffin bait technique, because this Nocardia utilizes paraffin as a sole source of carbon for its survival. So that's why this paraffin is required for its growth. So that's why the technique is known as paraffin bait technique. So again, it is a selective medium for cultivation of Nocardia. Okay. And what type of colony we will find in this medium? Waxy cerebriform or wrinkled colonies, or dry chalky white colonies. So you can see in this image this cerebriform waxy colonies, and if there is aerial hyphae formation, it will leads to dry chalky white colony as seen in this. So this is the characteristic of Nocardia species. Okay. Treatment. If you remember, the treatment of actinomyces I told you is through penicillin. But in case of Nocardia, the drug of choice is trimethoprim sulfamethoxazole, also known as cotrimoxazole, TMP SMX, and these are resistant to penicillin, so penicillin will not work on them, whereas actinomycosis, the drug of choice is penicillin. And for immunocompromised, we can use amikacin and cefotaxime, and for mycetoma we can do surgery to remove the lesions. Okay. So another question in this same topic is, a wound sample was obtained from a patient suspected to have nocardiosis. Which of the following method is best for isolation of Nocardia species? So among this option given, you can see the technique mentioned here is paraffin bait technique. This paraffin bait technique is used for isolation of Nocardia. This Castaneida's culture method is basically use of biphasic culture medium, and it is commonly used for typhoid fever and brucella. Okay, and this Craigie's tube method is used for bacterial motility. This hair bed technique is used to differentiate between various fungus. So the only option which is correct in this is paraffin bait technique. Okay. So I hope I was able to explain the topic to you, and it must be helpful to you for your examination. All the best. Take care.